and move to the second speaker, who is Ludovico Calabrese. Here he is. Ludovico, he graduated in physics in Milan at the State University with a thesis in statistical mechanics, statistical physics. He's now doing the PhD in the group of statistical physics of cells and genomes under the supervision of Marco Lagomarsino, still in Milan in the university, and also at the Institute of Molecular Oncology, IFOM. He is an expert in applying methods and concepts of statistical physics to study cell growth and gene expression. The title today is Macroscopic Models of Limiting Processes in Cellular Growth Laws. Please, Ludovico, you can start.
All right. So thanks for your introduction and thanks for the invitation. So yeah, I'm Ludovico Calabrese. I'm a PhD student. And as Federico was saying, I broadly use statistical physics to understand cell growth. So in this particular talk, I would like to talk a little bit about what limits cell growth and how understanding these limits can help us understand some macroscopic laws that govern cell physiology. So overall, I think that I hope that by the end of this talk, you'll be able to know a little bit how distinct growth limitation regimes can emerge in a complex growing system. So to begin with, I'll start with some introduction, and I'll start with some basic biological definitions. So of course, the first question is very simply just what is cell growth?
Ludovico, you have to share your screen. At least I don't see it.
You don't see it? Okay, I thought. How about now? Do you see it?
Okay.
Okay. All right, so you lost a little bit of that, but fortunately, let's hope that you didn't lose so much. I really just was starting with the introduction and starting to explain some basic concepts from biology that we needed to understand this talk. So as I was saying, the first question, of course, is what is cell growth? And by cell growth, we really just mean accumulation of mass and volume. So we're not focusing on how the cell divides, how the cell decides to split into two cells, but just into how the cells accumulate mass. And most of its mass, it's really protein mass. And therefore, to understand cell growth, it's really crucial to understand how the cell produces proteins. Of course, this is a very old question that was answered a long time ago, but I'll recap the basics. The basic way for protein production are encoded in the essential dogma of molecular biology, whereas from DNA, cells perform transcription using RNA polymerases and make mRNAs. They make transcripts. From the transcripts, cells perform translation with ribosomes, and they generate proteins this way. In particular, I want to highlight that RNA polymerases and ribosomes are the main molecular machines involved for performing these two steps, and we're going to be seeing them all throughout this talk. Generally, lots of these steps are highly regulated for expressing differentially different proteins. There are many caveats, I think, and several sub-steps involved here, but for the purposes of this talk, this is a pretty good enough picture, I think. So I think that on a microscopic level, we may say that cell growth or protein synthesis originates from large autocatalytic reaction networks. So you have a reaction network basically because each protein influences each other production, each transcript influences each other production. And in addition, such a network is autocatalytic in the sense that proteins catalyze their own production in some sense to have growth. So these types of networks have attracted the interest of the statistical communities, the statistical physics community in recent years. And such models can vary from very abstract ones to more biologically accurate ones. But overall, there are a number of steps involved that are always the same. For instance, people typically specify the topology of the network, as in this simple example with a single loop containing many different components. And in addition, people specify the form of the equation behind every link of the network. We're interested in cell growth, and therefore we're interested really in the growth of the overall size of the network, so considering all components. To characterize growth, we typically consider parameters such as this growth rate here, which is basically the logarithmic derivative of the total size of the network. So the growth rate, it's really a kind of intensive macroscopic parameters that characterizes the growth process, and we're going to be using this again massively throughout this talk. All right, this is in this field, people ask a number of questions such as what is the growth mode, whether it's linear, exponential, or some more complicated manner of growth. They ask what is the growth rate, as we've already seen. Another typical question is whether all components grow at the same rate, which is the notion of balanced growth. And a crucial one for us, it's also whether only some components of the network matter for setting the growth rate. And this is the notion of limitation regime, which is the one that we're going to be focusing on in this talk. So on a more macroscopic levels, people found empirically several regularities that characterize the behavior of cell growth physiology. And somehow these regularities must really emerge from the autocatalytic reaction network that we talked about before. So in this slide, though, I would like to talk about some of these regularities first, which have been called growth laws. So I would like to point out some general facts before going into some specifics. So you see three plots of such growth laws. And one important thing is that each point in these plots is obtained by growing cells in a particular medium with a certain nutrient quality. Higher nutrient quality means higher growth rate and faster growth. With each point, with each medium, people measure the growth rate and they measure the concentration of a molecule of interest. What's important, what's crucial really, it's that you can measure such observables in different medium, which may be very different chemically, but as long as they give the overall growth rate, the overall identical growth rate, you will have the same concentration. So what this means that effectively many details do not matter. And in fact, the growth rate effectively sets the concentration of many different molecules. So it's one parameter that sets really the concentration of many types of molecules of interest. So I would like to describe very briefly some of these patterns to give you a general flavor, and also because we are going to be using some of these. So starting from the plot on the left, you see on the x-axis the growth rate, and on the y-axis the protein concentration of a gene that is under no regulation, whether transcription or translation. There is really no regulation. And what you see is a decrease across growth conditions in this case. You might have expected that since there is no regulation, this concentration would be constant, but instead it goes down. In the plot in the middle, instead, you see that the total ribosome content increases with the growth rate, somehow suggesting that to grow faster, you need to produce more ribosomes. And finally, on the plot on the right, you see that the total mRNA concentration also increases with the growth rate, which also could be interpreted as saying that you also need to make more total mRNA. With that said, though, the first two plots here are relatively well understood, and there's a rich theoretical framework behind this, while this plot is much more recent, and people have not really reached a consensus on whether this means that mRNA is limiting for growth or not. Finally, before moving on, I'll point a final observation, which is that I've shown plots for this bacterium, Escherichia coli, but indeed some of these patterns have been seen for other organisms. For instance, in budding yeast, people have observed the same trend of the total ribosome content. This is striking because these organisms are very different. In fact, they come from different kingdoms with lots of different molecular details. But regardless of that, at this macroscopic level, you will see a very similar pattern, which suggests that there may be aspects of cell growth physiology that are really relatively universal. which is perhaps striking for a biologist, but it's the type of stuff that we physicists are interested in. All right, so I've shown you some examples of these growth laws. So let's, and I'd like to talk a little bit about what is the origin of these growth laws. What are the mechanisms behind these growth laws? And people have identified really two main concepts. The first one is the notion of a limited resources pool, a limited pool of resources. So imagine that within a cell, you have a finite pool of resources, a finite pool of machines, and some of these machines are allocated to production of a certain component, and other machines are allocated to the production of another component. So because the pool is finite, it will be the case that to upregulate the production of one component, you need to reallocate some of these resources to the production of a particular component, effectively stealing resources from the other component. So how does this apply in the growth context? Well, imagine that to grow faster, you need to produce a certain type of protein or a certain type of molecule. Because of the finite pool of resources, then upregulating this component will automatically decrease the production of other component. So for instance, the two growth laws that I've shown before are not really independent from one another, but they come from the same mechanism. In addition, you will note that to understand growth laws, therefore, it's crucial to understand what you need to produce to grow faster, which is really the notion of a growth limitation regime. So understanding what are the molecules that you need to increase to grow faster. I'll also note that while these two growth laws are pretty well understood within this framework, again, the mRNA growth law is not well understood in this context. So the final notion that I want to introduce is the concept of growth cost of protein expression. You see what this means on this plot. What it means, it's basically that if you overexpress a protein that effectively serves no purpose in the cell, you will observe a reduction in the growth rate. Growth laws help you understand why this happens. And this happens basically because by expressing a certain type of protein, you are in some sense stealing resources from all the proteins that instead are useful for growth. I also remind that the growth cost of protein expression is important because it helps you understand trade-offs involved between expressing a protein for some useful purpose and growth costs. So this is from an experiment. This plot, for instance, is from an experiment from 10 years ago. But more recently, people have performed such experiments with a slight twist. In addition to overexpressing such proteins, they also modulate the lifetime of the corresponding transcript. So transcripts, mRNA, has a lifetime, and which means that it basically degrades after some time. They observe that there is a striking change in the slope with this assay, and which seems to suggest again that perhaps mRNA also plays a role into setting the growth rate of the cell. So overall, what I wanted to say is that really most of the current models of growth laws focus on the translation remittent regime, where it's ribosome, it's the translation step of protein synthesis that sets the growth rate. Of course, theoretically, we know that there is transcription, so there must be in some sense a regime where transcription matters for growth. But in addition, there is growing evidence really from empirical data that transcription might play a role. So to conclude really this introduction, I'll formulate the questions that we are trying to answer. The first one is quite generally whether we can predict different limitation regime within a single unified model, especially trying to understand whether translation and transcription are limiting. And for doing that, we are going to be defining one of those autocatalytic networks that we talked about before. The second crucial question for us is whether transcription can affect growth and when. And our answer will be that there are two regimes where this can happen. And finally, we want to use our model to try to predict some of these unexplained growth laws that involve transcription. All right, so I'll now move to the results section. And I'll start basically with the first two questions that involve defining an autocatalytic reaction network model that includes both transcription and translation. So first of all, we define what are the nodes of our network. And the nodes are really all the abundances of the transcripts, first of all. So these are the mRNAs, and all the abundances of the proteins. Out of these transcripts and proteins, I wanted to single out here especially the proteins and transcript connected to ribosomes and RNA polymerases, because as we've seen, the ribosomes and RNA polymerases are the main machines that perform transcription and translation. So once we have defined the nodes, I'd like to move on to try to define the structure of the network, and in particular the topology here. I'll define it step by step in this slide. So you can imagine that you start from a particular protein, and to produce this protein, you will need the corresponding transcript, of course. In addition, though, you also need ribosomal proteins, because ribosomes are the machines that affect translation. Ribosomal proteins, of course, are no different. You also need the corresponding ribosomal transcript, and of course, they also catalyze their own production. Then you consider RNAP proteins, and again, you will need the corresponding transcript and ribosomal proteins to produce such proteins. But in addition, RNAP proteins are special within this network because they are the main enzyme that perform transcription, and therefore they are connected to all the other transcripts. So you can add nodes, you can add different nodes, of course, but the main structure of the network is really encoded in this diagram. We're now going to be moving on to describing the precise form of the reaction behind these links. And there are really two reactions. The first one is the translation reaction. So to produce a protein, you need mRNAs and you need ribosomal proteins. We use an initiation limited model of translation where a protein is produced in a way that the overall rate is proportional to the amount of transcript times an initiation rate that depends on the amount of free ribosomes. So free ribosomes have to bind to the transcript and then they start translation. So with further details and some maths, we can find that the amount of free ribosomes are connected to the overall concentration of ribosomal proteins, and therefore find an expression for this diagram. On the transcription side, things are somewhat simpler. mRNA is produced by RNA polymerases with a linear production term in our model, which is described here. And in addition, we also add a degradation term because, as we said, transcript can degrade, have a finite lifetime, and in fact, they degrade faster, and this degradation term cannot be ignored in standard physiological conditions. So given all of this, we start investigating the question of growth limitation regimes. And we identified three of them by looking on how the total quantities of our network grow with time. We find three regimes, and I defined them here. The first one is a ribosome limited regime, where the total protein production rate is proportional to the number of ribosomal proteins. The second regime is an mRNA limited regime, where the total protein production rate is proportional to the total number of mRNAs. And finally, we find a regime that we term a complex limited regime, where the total protein production rate is proportional both to the ribosomal proteins and to the concentration of mRNA. So it's complex limited in the sense that, we'll see in a moment. Indeed, I would like to give an intuition behind these three regimes. And to do that, rather than using a full model, which is a number of details that are not so transparent, I'll use a simple model coming from reaction kinetics. And I'll show you how this relates to our full model. So within this simple model of reaction kinetics, you imagine that you have two molecular species, A and B. A and B can form a complex with a certain binding constant. Once you have the complex produces a certain product P, and in addition, it dissociates into A and B. So you see that these are irreversible reactions. So in fact, this is a far from equilibrium model of reaction kinetics. So once you translate this diagram into equation, really just using mass action, you find a pseudo-phase diagram, so to speak, that can tell you what are the different limitations regimes with which this product is produced. Let's start from this region here. So when the total concentration of the two molecular species is below a certain threshold, you will find that the total product production rate is proportional to both the concentration of A and the concentration of B. Conversely, within these two other regimes, only one species is really limiting. And in fact, it's the species that it's present in the least amount. So if A is greater than B, it's actually B that sets the overall protein production rate. So we make a mapping then, conceptually, we can make a mapping between A, B, and P with mRNA, ribosomes, and proteins. And we can also then map the three limitation regime observed to the three limitation regime outlined in the model. So it's clearer now that this complex limited regime, it's really a regime where the time scale for forming the complex between the mRNA and the ribosome matter. So we use the model to answer one of the main questions that we said at the beginning. So when does transcription affect growth? So we found two regimes where this can happen. In the mRNA limited regime, since the total protein production rate is proportional to the overall number of transcripts. It is also possible to prove that within this regime, transcripts have to be spatially saturated by ribosomes. And in fact, through this idea, this regime was previously investigated in the literature. On the other hand, we also find another limitation regime, which is this complex formation limiting regime, where both ribosomal proteins and mRNA concentration matter for growth. And as far as we know, this regime is novel, at least in this context. In addition, we want to focus for the next part of the results on this regime, because we think that it's the one that best explains the data, as you'll see. And in addition, this condition of mRNA saturation is really quite extreme as far as we know, and it seems that it's not really the case in physiological conditions, at least for cells like bacteria. All right, so we'll move on to the final part of our talk and the last question that we wanted to answer, so whether we can explain some of the growth laws using our model. So we're going to be focusing first on the growth costs of unnecessary proteins. And to do that, it's relatively simple within our framework, because we can just use our reaction network and we add two nodes for the unneeded proteins, so corresponding to the transcript and the proteins of this unneeded gene. We then ask how growth is affected by them. So what we found is that theoretically, the growth cost is very different under different limitation regime. We're going to start by looking at what happens during the translation limitation regime. And you see that the growth rate decreases linearly as you increase the protein expression of this unneeded protein. In addition, when you change the stability of the unneeded transcript, you see that all the curves collapse to the same curve, which means that it really doesn't matter. Modulating the transcript lifetime really doesn't matter. On the other hand, you have a strikingly different behavior when you consider the complex formation limiting regime, where both ribosomal proteins and mRNA matter. Here again, you see a linear dependency between the relative growth rate and the protein fraction. You see a decrease, but the slope now changes dramatically when you change the lifetime of the unneeded transcript. So we wanted to also look at some data on this, and indeed, as I've shown you before, there were data on this. So we used this data set from budding yeast. We are able to fit the parameters of the model in the stable condition where the unneeded transcripts are relatively stable. Experimentally, then, they are able to tune the stability of the transcript, and we can use the model to predict how the relative growth rate changes under mRNA stability changes. You see that when you use the translation limitation model, the relative growth rate, the shape of the relative growth rate against the expression level remains the same, so you cannot explain this change in the slope. On the other hand, with the complex formation limitation regime, we can capture the change in the slope precisely. All right. So to conclude with, we wanted to also look at the mRNA growth law that we've talked about in the introduction. To cut a long story short, we find that our model can predict this behavior, this mRNA growth law, by optimizing the growth rate within the regime of complex formation. The story is a bit more complicated, actually, because in the simplest version of the model, we actually find by this optimization procedure a square root growth law. So the mRNA concentration goes with the square root of the growth rate. And to get a linear law, as the one that we see in the data, we need some further element. And we think that this further element is an increase in transcription efficiency as well, which is what it's seen in the data as well. All right. So really, I think that I can wrap this up. I'll summarize our main conclusions. So first of all, we define a general mean field and non-equilibrium model of biosynthesis through this autocatalytic reaction network that involves both transcription and translation. With this model, we can identify different growth limitation regime, and in particular, we can answer the question of when is transcription limiting. We found two regimes, and especially one macroscopic mRNA-dependent growth regime that is due to complex formation, the ribosome mRNA complex formation. Finally, we can use this regime to predict some of the unexplained growth laws of transcription across different organisms, which again, it seems to suggest that there are some universal aspects of growth physiology, since the models can be applied to really different organisms that live across kingdoms. All right, so this is the end of my talk. So I thank everybody, of course, for their attention. I thank my group, and specifically my supervisor, and of course my collaborator here, Luca Cindrini. So if you have any questions, of course, ask away.
Okay, thank you, Ludovico. The session is open for questions, if there is any. No questions. Maybe I have a comment. It seems that this mean field description works well, at least up to some level. So as far as I can understand, mean field could be good because inside the cell you have all these elements that in some sense they move, so they get in contact with each other. Is this correct, or there is something different?
Yeah, I think there are a couple of things there. First of all, I think that mean field, in the sense of not considering a stochastic version, for instance, with Markov models, works here because really we are considering a cell that is packed with 10,000s, if not more, of these components, right? So the mean field approximation works quite well, I think, there. In addition, I haven't said this, but we could, in principle, change many of these parameters for different mRNAs and so on. Generally, we have assumed that many of these parameters are homogeneous. For instance, say that the degradation rate of the transcript here is the same for all types of transcripts. We think that this further heterogeneity does not really affect the overall conclusion of these growth limitation regimes, basically. So I hope that I somehow answered the question.
Yeah, more or less. And these parameters, they are taken from some experiment, or how do you fix them?
Right.
This k omega.
Sure. So it depends. It depends on the question. For some questions, these parameters don't matter very much. For instance, when we consider just the relative growth rate, for this case, these parameters just don't matter for formulating this prediction. In other cases, yes, we either fit them or we can of course extract them from the literature when they are present. But I think that the crucial thing is that some of these predictions really are not dependent on the particular number associated to those parameters.
Hopefully, yes. Thank you. Any other questions? Okay, if there are no questions, I think we can close the session. We meet again after 1 month. More precisely, let me have a look. It should be on the 13th of April. Okay, bye bye everyone. Ciao a tutti.